Okay, our next presenter is Jim Reid, CEO and Chairman of Sistemic. Jim.
Thanks, Rob. Yeah, I'm Jim Reid, Chairman and Chief Executive of Sistemic from Glasgow and Boston, and no prizes for guessing which picture is which, and no points for guessing that I'm also from Scotland. I'm based there. But somebody told me today I'm wearing a Giants tie, which really didn't mean anything to me. I guess it's the colors. So anyway, let's see if we can move on. We are the microRNA cell profiling company. I hope you're beginning to hear about us. And our mantra really is that every cell tells a story, and what's yours saying? We believe that people really need to understand precisely their cells before, during, and immediately before any use in cell therapy. So the more you know about it, the more efficacious it's going to be, the lower your costs are going to be in production, and the better outcomes there's going to be for the patients. And using microRNA, we believe, is the way for this to happen. In terms of the corporate overview, we're a VC-funded company established in 2009. We have a broad IP portfolio in around microRNAs for profiling, but in context, i.e. understanding the biology. We have HQ and labs in Glasgow and a sales office in Boston, and hopefully shortly a sales office here on the West Coast. We have currently 10 staff, and we expect to increase to 16 during 2013. We have a two-strand business model at the moment where we're looking at drug discovery and cell profiling using our SistemQC service concept. And I'll explain to you a little bit about the dynamics of our business as I go through. It's multi-structural, i.e. it's service-based, moving to licensing, and eventually into products. In terms of SistemQC, it's a suite of cell characterization and QC monitoring tools based on robust microRNA profiling, where we look at the entire microRNAome. We also bring superior statistical analytics to identify what we call the key microRNAs, or kemers, which allow identification and assessment of phenotype. We look at molecular characterization linked to the biology, and this is vitally important. And we use that to look at the kemers from a multi-layer contextual analysis point of view. So we want to really get to the detail about what's happening. And the next slide will show why we think it's important. This is a comment from Eric Lander, director of the Broad Institute in Boston, where he said, "Some scientists we managed to miss an entire class of incredibly important regulatory molecules until the 21st century." And this is the absolute critical thing. And I'm going to take you guys back to first-year genetics, where we're looking at the microRNAome sitting between the genome and the environment and really managing the interpretation to result in the phenotype. So the old statement of phenotype equals genotype plus environment is underpinned by what's happening in the microRNAome. And that's the critical thing as far as we're concerned. And this further illustrates the situation where the microRNAome sits at the pinch point between the transcriptome and the proteome. And we believe by interpreting and understanding precisely what's happening here, people can really understand precisely what's going on within their cell populations, because these microRNAs are micromanagers of gene expression. They're cell and tissue specific, and they're fundamental in cell fate. Analytically, they're very stable. They're present in all cells, analytically robust. It's a very manageable data set. There's approximately 1,300 or so microRNAs in MIR-base 16, which we're using now, 1251. And they're present in the exosome, so it's suitable under certain circumstances for non-invasive analysis. And the interpretation of that really allows one to describe very precisely the phenotype and understand the relevant biological characteristics. Okay. So in terms of the characterization and QC, we can, and I'll show you some data on this, but we are essentially looking at sophisticated characterization for identity, potency, purity, optimization of differentiation strategy, and cell optimization and selection, i.e. process efficiency. And in terms of monitoring, looking at cell or process change, i.e. comparability, batch-to-batch consistency, and benchmarking of cell model systems. So all of this comes together under the SistemQC banner. And what we're doing is using our analytical approach to generate keyers. We're looking to prioritize these analytical properties and give people a progression through prioritization, verification, validation, and utility, all underpinned by our corporate IP. And we want to look at maximizing that through novel markers and background, but always with an understanding of what the regulators need. And we take this issue very seriously. There's no point in us developing something which subsequently puts a red flag to the regulators. So we engage with them, we talk to them, and we're very serious about this particular issue. And just quickly to move on to some data, and I thank our colleagues at Labs Inc. for this data. In this one, we're looking at unique ID markers for similar cells, and here we see 3 populations of MSCs. And we present this schematically in a PCA, a principal components analysis plot. It's simply so as you can visually look at the differences between the cells. And what we do is we reduce that to practice where we look at the actual key microRNAs which allow that differentiation. And here we have a panel of less than 4 validated ID keyers which can then be used routinely using qPCR for in-house testing. And these keyers are aligned with the biological phenotype. Okay. You can see. Moving on, and again, I thank our colleagues at Rosalind Cell for this data. And here we see the expansion of HESCs, we're looking here at results which show that on flow cytometry, these cells are essentially the same. And even after high pass-size numbers, there are no difference in terms of the flow cytometry markers. So one might conclude that these cell populations are the same, but using microRNA profiling, you can see that they are dramatically different. So the microRNA SistemQC looks at the differences between what were classed as identical pluripotent population. And these microRNAs we subsequently studied to look at the biological link. And that's shown on the next slide here. And you can see that cell differentiation is a major, is a major part of what's going on here. So clearly these cells are not the same. And what we're able to do is we can say that we're identifying profile drift. We're looking at the pathways and processes that look at that drift. We're looking at differentiation with increased passage number. And we provide the signature, which can then be used as a routine monitoring tool. A little bit about our business, and you'll understand why we're interested in the cell therapy space. If I would have shown you this slide 1 year ago, we would have probably seen 50% of our business in drug discovery and 50% of our business in stem cell therapies, or QC. Now we're seeing hugely, almost 90% of our business in the cell therapy space. So people are realizing the power and the value that microRNAs can bring. Geographically, and the reason why we're interested, of course, in being here, is that about 60% of our business is in the US. And that's why we have an office in Boston and why we're looking to put an office in the West Coast. But we like to think of ourselves as being global. We have business in Korea, Australia, Japan, and Europe. So we think of ourselves as being a truly global company. In terms of our corporate pipeline, and I realize that people are used to looking at therapeutic companies with phase 1, phase 2 trial information, etc. We're slightly different. As I say, we were set up in 2009. We entered our development phase there. We introduced our first service products at the beginning of 2011. We're expecting to see the first license partners at the beginning of 2013, and the first products coming through in 2014. So we have a transitional business model where we want to work with partners to look at their profiles, to help them identify identification panels for their particular cell lines, to then transfer that technology to them to use to give them in-house utility under license. And then at some point in the not too distant future, 2014, we're forecasting to start to produce a range of routinely usable products which allow characterization of different stem cell populations. And just quickly, I want to show you that we are truly global. This represents, each star represents a client that we're working with. So predominantly West Coast, East Coast, Europe, Korea, Japan, and Australia. And I want to thank you for the opportunity to present today. I want to say that I'm here for the entire period. My colleague, Werner McKerlin, who I'm sure many of you know, is here also for the entire period. We have a booth on the platform outside, and we're available if anybody wants to talk and learn more about SistemQC and what Sistemic are doing as a company. Thank you very much indeed.